Hi everyone, welcome to today's lesson on Sn1 and Sn2 reactions. This lesson is adapted from the PSHS Chemistry 3 Module Lesson 2.10 and 2.11. For this lesson's goals, first we describe the unimolecular nucleophilic substitution reactions Sn1 and bimolecular nucleophilic substitution reactions Sn2. Geraniol is an alcohol extracted from roses. It's used in perfumery and cosmetic products and has antimicrobial, antioxidant, and anti-inflammatory activities. Biosynthetically, it is produced by two Sn1 reactions. To get a closer look, first, we try to understand what nucleophilic substitution reactions are. So nucleophilic substitution is a polar reaction where a functional group in the electrophilic substrate is replaced by the nucleophilic reagent. So here we can see the nucleophilic substitution reaction where we initially have a substrate with a leaving group being attacked by a nucleophile, producing the detachment of the leaving group from the substrate and its replacement by the nucleophile. There are two types of nucleophilic substitution reactions. First is Sn1 and the next is Sn2. So first, let's take a closer look at unimolecular nucleophilic substitution or Sn1. S in this case means substitution, N means nucleophilic, and 1 for unimolecular. So of these three terms, first, let's understand what each of them means. So first, it's a substitution reaction, meaning that the leaving group is replaced in the process. Also, it's nucleophilic, which means that what substitutes the leaving group is actually a nucleophile or a structure which is either electron rich or at least attracted to a positive entity. And then finally, it's unimolecular, which means that it follows first order kinetics. That means that the rate determining step only depends on the concentration of one species. In this case, it's the substrate. So here is the reaction mechanism for Sn1. First, we have this substrate where the leaving group detaches from the alpha carbon to form a carbocation intermediate. So here we can see the substrate with the leaving group detaching itself from the structure, producing a carbocation where the carbon in this case only has 3 groups around it. In the second step, the nucleophile attacks the carbocation intermediate and replacing the leaving group. So in this case, we have the carbocation with 3 groups around it being attacked by the nucleophile, resulting to a final structure where the alpha carbon finally has 4 groups around it. So in terms of the shape, notice that our initial substrate has a tetrahedral shape with 4 groups around the alpha carbon. And then after the leaving group has left, then we have the carbocations intermediate, which is trigonal planar because there are only 3 groups around the carbon. And then after the nucleophilic attack here, we have a final shape, which is tetrahedral. Another important feature of the Sn1 reaction is the possibility for either inversion or retention. So in this case, after the leaving group has left to form the carbocation, notice that the nucleophile could actually attach itself or attack the carbocation from whichever direction possible. So if it attaches itself from this direction, then we can produce a structure that would look like this or oriented in this manner. Notice that there is an inversion between or comparing the substrate and the product. Or in the other case, we may also have the nucleophile attacking the carbocation from the same direction, therefore producing a final product with the same configuration or the same orientation in space as the substrate. And therefore, this is considered retention. Now, these two products being formed by the Sn1 reaction may be considered a racemic mixture. That is if this carbon or the alpha carbon is the only chiral center in the structure. And the process of producing both an inverted configuration and a similar configuration in the products is called racemization. Again, because the Sn1 reaction produces a carbocation which may be attacked by the nucleophile on either direction, which may result to an inversion or a retention in the final configuration of the product, then racemization occurs. So that we can see better the shapes and the 3D view of these structures. So let us take a look at these illustrations. Here, let's consider the substrate having the tetrahedral shape. And then the first step in an Sn1 process, again, is the detachment of the leaving group, forming this structure, which is a carbocation in this case. And notice that the alpha carbon, instead of having 4 bonds around it, now only has 3 groups around it. Therefore, it will take on a new shape, which is trigonal planar. Okay, so that is the 3D view of these species. We have the tetrahedral shape here, and after the leaving group has left, we will have a trigonal planar shape. In the second step, again, we start with a trigonal planar carbocation. Notice that, as I have said, the nucleophile may attack this structure from either direction, therefore producing 2 possible configurations for the products. That would be either attaching itself on this side or on the other side, producing 2 different shapes or orientation in space or configuration. In terms of energy for unimolecular nucleophilic substitution reactions, so let's take a look at this graph. So as the reaction progresses, the energy of the reaction mixture follows this pattern. So initially, the reactants would have this level of energy, and then as the reaction progresses to form the intermediate, so here's the level of energy, And then notice that from the reactants to the intermediate, there's a high energy species here, which would correspond to the transition state. And the energy difference between the reactants and the transition state would be equivalent to the first activation energy. And then finally, the products are more stable than the reactants. So that would correspond to the substituted substrate. And then also notice that there's an energy difference associated with the intermediate, between the intermediate and the second transition state. So that would be equivalent to the second activation energy. So notice that the activation energy for the first step is greater than the activation energy for the second step in the reaction. It means that the first step is considered the slow step, which is rate determining. Okay, so here we have two transition states. So the structure for the first transition state would be intermediate between the reactants and the reaction intermediate. So in this case, the transition state would have a structure where the leaving group has partially left the structure. Or in other words, this bond between the leaving group and the alpha carbon is partially broken. So that's what the high energy transition state structure would look like. On the other hand, for the second transition state, its structure, again, would be in between the intermediate and the product structure. Therefore, we can say that in the second transition state, the alpha carbon to the nucleophile bond is partially being formed. And that results, again, to a high energy state. Next, let's move on to bimolecular nucleophilic substitution reactions, or Sn2. So Sn2 reactions, again, are still substitution reactions where the leaving group is replaced, and what replaces them is a nucleophile, which is responsible for the attack on the substrate. And finally, it's bimolecular, which means that it follows second-order kinetics, meaning that the rate of the reaction is dependent not only on the substrate, but in this case, both on the substrate and the nucleophile concentrations. So here's the reaction mechanism for Sn2 reactions. First, notice that we have the substrate and the nucleophile here, and the whole reaction occurs in one step only. So there's a simultaneous attack of the nucleophile on the alpha carbon and detachment of the leaving group. So all in one step, the nucleophile attacks the alpha carbon, and because of that attack, the leaving group also leaves almost simultaneously. So do not be deceived by the two arrows here, because this structure is not a reaction intermediate, rather it's the transition state. So this whole mechanism is only just one step. And if in the previous reaction, Sn1, we did not illustrate the transition state anymore, so here in Sn2, we can see the illustration for the transition state, which is actually a pentavalent structure, meaning there are 5 groups around the alpha carbon. Notice also the bond that is partially broken with the leaving group and partially formed with the nucleophile, being represented by dotted lines here. And because of this process, The only possible orientation that the nucleophile can take in order for it to attack the substrate would be opposite the leaving group. Therefore, emphasis is given on this structure where the leaving group leaves in the opposite direction compared to the direction of attack of the nucleophile. Therefore, after the reaction, there is complete inversion. So the leaving group leaves from this side while the nucleophile attaches itself on this side, resulting to a complete inversion in the alpha carbon configuration. In terms of the energy as the reaction progresses, so notice that there's only one leap in the energy diagram corresponding to just one transition state, also corresponding to just one step in the process. So again, this level would correspond to the energy of the reactants, and then this energy would correspond to that of the transition state, which is very unstable and only exists in the mixture, maybe in a split or a fraction of a second. And finally, the level of energy of the products, which is, as you can see, of opposite configuration, or which is a result of complete inversion. So let's take a look again at the shapes or the 3D view of all of these species. Here we can see the tetrahedral structure of the substrate. This is the leaving group and this is the nucleophile. So when they react, they're going to form the pentavalent transition state, which I tried to represent in this manner. So here's the alpha carbon, and then here's the leaving group partially leaving, and here's the nucleophile partially attaching itself to the structure. And because there are 5 groups there around carbon, so it's considered pentavalent, and it's going to have a trigonal bipyramidal shape. Also notice that the leaving group and the nucleophile would be oriented in opposite poles or as far away from each other as possible. After the reaction, notice that the leaving group has successfully left the substrate and the nucleophile has completely attached itself to the alpha carbon, and there has been an inversion in configuration. And finally, the alpha carbon resumes to the original tetrahedral shape around its structure. That's it for the bimolecular nucleophilic substitution, or SN2. So next, we're going to compare SN1 and SN2 reactions. So here, we have the reaction mechanism for the first step in the SN1 reaction and the reaction mechanism for the whole of the SN2 reaction, which occurs just in one step. So comparing the rate law, SN1 is unimolecular and its rate depends only on the substrate, because in the first step or in the rate determining step, notice that there are no other reactants except for the substrate, which first forms a carbocation. Therefore, its rate law is only dependent on the concentration of the substrate. On the other hand, for the Sn2 reaction, the first step involves the immediate attack of the nucleophile to the substrate, producing this pentavalent transition state. Therefore, its rate law is bimolecular, and the rate is dependent on both the concentrations of the substrate and the nucleophile. Next, in terms of the big barrier means the greatest concern of the reaction. So for SN1, we're mostly concerned with carbocation stability. So the more stable the carbocation, the faster the rate determining step happens. On the other hand, for SN2 reactions, we're more concerned with steric hindrance. That means the space available for the nucleophile to occupy or to attack or to perform this backside attack to the substrate. Therefore, that would also dictate how alkyl halides are more preferred, or which alkyl halides are more likely to produce or to undergo SN1 or SN2. So for the SN1 reactions, tertiary alkyl halides are more likely to undergo the reaction than secondary alkyl halides and primary or methyl alkyl halides. So why is it that tertiary alkyl halides are more likely to undergo SN1? Again, that's because of the fact that the first step in an SN1 reaction is the formation of carbocations. Therefore, tertiary alkyl halides produce more stable carbocations and are therefore occur faster compared to primary alkyl halides. On the other hand, for SN2 reactions, primary alkyl halides have the least steric hindrance compared to secondary and tertiary alkyl halides. So here, since the first step involves the attack of the nucleophile to the alpha carbon, then it would be more preferable for the alpha carbon to be less hindered around it. Therefore, a methyl alkyl halide or a primary or a secondary would fare much better than the crowded tertiary alkyl halides, which would leave no space for attack by the nucleophile. Therefore, for tertiary alkyl halides, an SN1 reaction would most likely occur, whereas for primary alkyl halides, SN2 reactions are more likely to occur. For the nucleophiles, weak nucleophiles are generally preferred by SN1 reactions because in this case, the nucleophiles would only need to attack the carbocation after it has been formed. On the other hand, you would need a stronger nucleophile for the SN2 reaction because it would take active part in pushing away the leaving group. So again, a weak nucleophile would favor an SN1 reaction, while a strong nucleophile favors an SN2 reaction, where it plays a much vital role in pushing the reaction forward. So you might wonder what's a strong nucleophile and what's a weak nucleophile. So here are the strong nucleophilic species. Notice that strong nucleophiles are highly negative structures, meaning they attract or they are more attracted to positive structures, positive charges, partial charges. On the other hand, weak nucleophiles are in general also polar structures, but not very strongly negative. Also, in terms of the solvent system where the reactions are taking place, so SN1 reactions prefer polar protic solvents. Polar protic means that they're polar, meaning there are partially positive ends and partially negative ends to the molecule. Protic means that they're able to solvate protons or H plus or hydronium ions in the system. So in this case, polar protic solvents are advantageous because they're able to dissolve carbocations better. So when the carbocations are more stabilized, this reaction becomes faster. On the other hand, for SN2 reactions, polar aprotic solvents are preferred because aprotic solvents are able to on the other hand, for SN2 reactions, polar aprotic solvents are preferred because they do not dissolve the nucleophile too much, therefore allowing it to react more potently towards the substrate. In terms of stereochemistry, again, SN1 reactions produce racemic mixtures or result to a mixture of retention and inversion in terms of configuration. On the other hand, for SN2 reactions, there's complete inversion. Let's try answering the following questions. Which among the following solvents promotes SN2 reactions and which ones promote SN1 reactions? First, we have dimethyl ether. What do you think? Dimethyl ether is an aprotic solvent, therefore, it promotes SN2 reactions. Next, we have dimethyl sulfoxide. Again, it's also aprotic, therefore it promotes SN2 reactions. Finally, we have water. So we know that water undergoes autoionization, producing OH minus and H plus or hydronium ions in the system. Therefore, it promotes SN1 reactions. Next, let's determine whether the following nucleophilic reactions will most likely proceed through an SN1 or an SN2 mechanism. So let's look at this substrate. The alpha carbon of the substrate is actually secondary, and the solvent is protic. Therefore, this will most likely proceed as an SN1 reaction. Next, we have a substrate with the alpha carbon, which is primary, and the solvent, in this case, is aprotic. Therefore, it's most likely going to proceed as an SN2 reaction. So that ends the lesson for SN1 and SN2 reactions. Again, we were able to discuss how SN1 and SN2 reaction mechanisms proceed and their differences. I hope that this video has clarified your questions about the topic and helped you understand the lesson better. So that is all for this video. Goodbye, class.